Hi everybody, I'm JP Johnson. I'm from Xenon Pharmaceuticals in Vancouver, and we've been around a little bit longer than Sophion. We started in 1996. We still do not have a product on the market. We hope to change that soon. Our lead project is a Kv7 potassium channel potentiator, which is now in phase 3 clinical trials for focal onset seizures, so we hope to have that to patients with refractory epilepsy soon. But today I'm going to talk about an earlier stage project that's potentiators of Nav1.1 channels for Dravet syndrome. For people who are not familiar with Dravet syndrome, it's a really severe developmental epileptic encephalopathy. It's usually caused by mutations in SCN1A, loss of function mutations, which causes a reduction in the number of Nav1.1 channels in those patients. So it's usually a de novo mutation. These patients usually have about half as many Nav1.1 channels as they ought to have. And that causes lots of problems, including severe epilepsy. Children are born appearing healthy, they seem good, they're meeting their milestones, and then about the age of 6 months, when normally SCN1A expression would be increasing, it doesn't increase enough in these patients, and so they end up with a deficit of Nav1.1 channels, and that leads them to seizures. It usually presents as status epilepticus in the emergency room, and that's the first sign that parents have that there's a problem with their child. After the seizures begin, the problems multiply. There's lots of other developmental problems, behavioral problems, movement disorders, lots of problems that these kids have. And in fact, about 15% of these kids will die before they reach adulthood. So it's a really, really severe epileptic syndrome. And the kids need a lot better treatment than they have available. Fortunately, we do have a mouse model that works pretty well for this disease, or it seems to. It's like the patients, these mice are deficient in Nav1.1. They are missing a functional copy of of the SCN1A gene. So they're deficient in Nav1.1. Like the patients, they are born pretty healthy. And then at about 3 weeks of age, about postnatal day 21, they start to have seizures. And in the next 2 weeks or so, between P21 and P35, about half of these mice will die from their seizures. They also have other features of the disease, like impaired motor function, cognitive problems. And we got these mice from Jennifer Kearney's lab, where they were developed. A lot of other people, a lot of other groups have used these mice as well. So why does losing Nav1.1 cause such big problems? It seems that the primary problem is that there's a reduction in the amount of Nav1.1 in inhibitory interneurons. So these, specifically the parvalbumin positive, rapidly firing interneurons, when you don't have enough Nav1.1 in these neurons, they become hypoexcitable, and as a result, they don't release enough GABA onto the primary glutamatergic neurons, and those neurons are disinhibited. They fire too frequently, and they synchronize, and you end up with seizures and lots of other problems. So our goal was, these patients, they still have half as many Nav1.1 channels as they ought to have. There's still some there. Let's focus on the channels they have and turn up the function on those, and then we can rebalance the system. But as you can imagine, selectivity is really critical for an approach like this. All the other sodium channels that are very similar to Nav1.1 are potential problems, right? Nav1.2, Nav1.6, these are the most critical sodium channels in the primary neurons, the glutamatergic neurons. And so if you turn up the function on those, you're going to make those neurons more excitable, you're going to exacerbate the problem. We don't want to do that. If you potentiate Nav1.4, you'll have muscle problems, 1.5, cardiac problems, 7, 8, and 9 all are likely to cause pain. Okay, none of those are good scenarios, so we really need to have compounds that are very selective for Nav1.1. Fortunately, we have some really strong medicinal chemists who have solved this problem for us. So we now have compounds that very specifically upregulate Nav1.1. So you can see here a concentration dependence on the right, increasing the level of XPC837 leads to more and more potentiation of Nav1.1, but with the Nav1.2, 6, 5, there's really no effect, right? And we're showing 2, 6, and 5 here because we think these are the most important targets that we want to avoid. But in fact, it spares all the sodium channel isoforms with the exception of Nav1.3, which is largely an embryonic channel. So we think that it's okay that the compounds potentiate Nav1.3. So the way that this potentiation is working is by inhibiting the channel's ability to inactivate. So if you look here on the left, you can see a normal looking sodium current in black, which rapidly activates and rapidly inactivates. You have very little persistent current. But when you add XPC837, you begin to get persistent current because the channels are not fully inactivating. And more X837 means more and more persistent current. And the way that it is affecting that, the inactivation is by shifting the voltage dependence of inactivation. So if you look at a classic H infinity curve here in the middle, where you're looking at the voltage dependence of inactivation, in black is the control cells, and I should point out that this is all data from the Sophion cube. So in black is control. When you increase the concentration of the drug, you shift the voltage dependence to the right. So that at saturating voltages, you have about a 30 millivolt shift in the voltage dependence of an inactivation. So at voltages where normally the channels would be inactivated, they are no longer inactivated, and they can still pass sodium current. You have more sodium current flowing through the same number of channels. And we hope that that will have a beneficial effect in the patients. So once we have these compounds that very selectively potentiate Nav1.1, we want to see what do they do in neurons. So here we're looking at brain slices from a dravet model mouse. So these are mice, again, they're deficient in Nav1.1, and that leads to hypoactivity of these interneurons. So we're selectively looking here at parvalbumin positive, rapidly firing interneurons in the slice. And when you look at one of these neurons, what you see is that they will fire very rapidly. In a healthy mouse, these neurons will fire at about 100 hertz, and they'll continue to fire at about 100 hertz in the face of a continued stimulus. They're very fast firing, but in these Dravet mice, they don't continue that rapid firing. They're prone to depolarization block. And so if you give them a stimulus, they'll start out firing quickly, but then the firing will drop off and they'll go quiet. When you add 837 to these neurons, you can see that you restore firing. So this is the same neuron before and after treatment with 837, just in a slice, in a dish. And here it's 250 nanomolar. So if you look at a classic kind of input-output curve where you inject more and more current and you count how many action potentials are happening in this 1 second stimulus, you get more and more, and the black line is what would happen in wild type. You get more and more until you get to about 100 hertz, and then you plateau. But with the Dravet mice, those neurons, they start out to fire rapidly, but then when you push them too hard, they go into depolarization block. The number of action potentials drops off. But when you treat that slice, again, with 250 nanomolar 837, you restore the firing, and they continue to fire at that rapid rate like a wild-type neuron. So one thing about these kind of slice experiments is you never know exactly how much drug you're getting into the slice. You can't assume that you're getting the same concentration in the slice that you're perfusing. So we actually measure that after the experiments. We homogenize the slice, and we quantitate how much drug is in in the slice. And some cases there's less, and some cases there's more than what's in the bulk solution, depending on the compound that you're looking at. This compound accumulates a little bit in the slice, so in the slice at the end of the experiment, it's actually about 400 nanomolar, even though 250 nanomolar was applied. I'm going to skip that in the interest of time and move on to the part that people are most interested in, which is, what does this do in a whole animal if you give these compounds to mice? So one fundamental challenge of pharmacology in rodents is that they're champion metabolizers. They really break down the drug very fast, and that's often true, usually true. Generally, drugs are much more stable in humans than they are in rodents, and mice especially so. So if you give a mouse an oral dose of drug, it will be on board for an hour or two, and then it's rapidly eliminated. So if you want to do long-term experiments where you're looking, say, at spontaneous seizures, for example, that's a challenge. You can't really do it with just a simple oral gavage kind of approach. So to get by that, we have incorporated the drug into the food of the mice. And so what we do is these mice are born P0. On P19, we wean the animals, we introduce solid food, but just control solid food, normal food. And then on day 21, we randomize the mice, and half of them will continue to get normal food, half of them will get food that has drug incorporated. And then we look at how the two groups do. And what we see is that, as I told you, after about day 21, these mice will start to have spontaneous seizures. It varies a little bit from batch to batch. You can see in this group of animals, the seizures started more like day 26, and they increase over the next few days and until they kind of plateau at day 33 or so. And this is typically what you see. Sometimes the seizures start a little earlier, sometimes a little later. But when we incorporate the chow, the mice that have been on 837, those seizures don't really take off. You can see that we had a couple of seizures very early in the experiment, and then they just stopped. And the mice are no longer having seizures. We did dial this in. It took some iterating, but we dialed this in to get an exposure in the brains of the mice. So these are all quantitated after the experiment. When we sack the animals at day 35, we measure the concentration in all their brains. And the concentrations we're seeing here to get this kind of response is just about exactly what it took to correct the firing of those interneurons in the first experiment I showed you. So here we're at about 500 nanomolar as opposed to 400 nanomolar in the slice. And that protection also extends to survival. So as I said, about half the mice typically die in an experiment. It does vary somewhat from group to group, but here you can see in this group of animals, about half of the vehicle exposed animals died, and when we had 837 on board, we lost one animal very early in the experiment and then no more. So the protection is not only the seizures, but also the animals survive as well. As I mentioned, the seizures are, of course, a big problem, and we were very excited to get this data. But what we really want is to have a broader impact on patient health, and not only to stop the seizures, because some of the non-seizure issues that these patients have are even more disruptive to the patients and their families' lives than the seizures are. So we wanted to look to see whether other features of the phenotype was improving. And so here's an example looking at a rotor rod. So this is a spinning rod. You put the mice on it, and they walk or run to stay on the rod as long as they can until they eventually fall off. And with a healthy mouse, they stay on the rod about 60 seconds or so before they fall off. And these dravet mice, they're clumsier. They can't stay on the rod as long. And so they average more like 40 seconds. But you'll notice here that it's pretty diverse. Some of them, the dravet mice, are pretty coordinated. They do well. They stay on as long as a wild-type mouse, and whereas others are clearly impaired. So what we do to kind of separate the signal from the noise is that we identify these animals that are underperforming, these impaired mice, and we take that group and randomize them into either to get drug or not drug. And what you can see is that the animals that didn't get drug, when they're tested again, perform worse than the animals that did get drug. So it looks like the motor control is actually improving in these dravet mice treated with compound. Now this is a single dose of compound. There's only one dose. It is not the chronic dosing that you saw in the survival studies for technical reasons. But it does seem like it's improving the coordination. And then we went on to look at other features. So we started to look at the structure, actually, of the neurons in the brains of these mice. And so Managaza's lab, Massimo Managaza's lab, recently did a really nice study where they showed Golgi staining in slices of dravet mice. And what they found is that the dendritic structure is different in the dravet mice versus the healthy mice. So in dravet mice, there are more of these long, thin spines shown here on the left of this cartoon. And as the spines mature, they move towards more of a short, stubby mushroom shape. And so the dravet mice, they showed, have more of the long, thin neurons, or dendrites, synaptic spines rather, and fewer of the stubby mushroom-like spines. And so these more mature spines are thought to have stronger synapses. They're stronger synapses. They're more efficient at passing signals. So when we see in our animals exactly the same thing that they saw. The wild-type animals have fewer of the long, thin, immature spines, and they have more of the stubby or mushroom spines than the dravet mice. But when we treated the animals for 2 weeks on the 837, you restore that wild-type-like profile. You have more mature spines, fewer of the long, thin, more of the mushroom spines, and so we think this shows that we're not only are we stopping the seizures here, but we're improving or normalizing the mice even at a structural level of the neurons. So if the neurons are more mature, the spines are more mature, the synapses are stronger, you should be able to read that functionally. And in fact, we can. So what we're doing here is slice experiments in looking at long-term potentiation. So if you're not a LTP person, then the way you do this is you stimulate in one part of the brain and you record in another part of the brain. And the signal that you measure at the remote site has to pass through multiple neurons, multiple synapses to get to the remote site. So the size of the signal you record there is a indicator of how strong the synapses are between the two locations that you're recording from. And so if you are doing these kind of recordings and you do a really high frequency stimulus, like theta burst stimulus, you see a potentiation of the signal is a short-term potentiation, which then decays into what's known as the long-term potentiation, or LTP. And you can see that the wild-type mice have a larger LTP signal than the dravet mice. So the dravet mice are impaired in their long-term potentiation. And this is thought to be a marker for learning and memory. The mice that have been on 837 for 2 weeks through their chow don't look like they're their dravet siblings, their untreated dravet siblings. They look more like wild-type mice. So you've restored the long-term potentiation. So what we think is that that means not only we're going to stop the seizures, but we're going to globally improve the function of the brain, at least in these mice, and hopefully in patients. So the state of the art for Dravet patients is really just seizure control. All the drugs that are available are focused on stopping the seizures, and they can be pretty effective for some patients. Still about 90% of the patients are still having some level of seizures. And they're not really getting that much benefit beyond the seizure control. There are new therapies that are in the clinic, like genetic therapies, for example, from Stoke Therapeutics. They have had some really interesting success with an ASO-based approach, which reduces the seizure count and also is showing some really promising signs of improving communication and coordination in those patients. So that's really exciting for us, but we don't think that, or we think that our compound could do even better. One challenge with the Stoke approach, the ASO approach, is that you have to inject it directly into the spine every few months. Obviously that's, kind of a hassle for the patient and the families. But also there's somewhat of a distribution issue where you can, it's hard to get to the deep neurons in the brain, and our compounds being orally dosed and brain penetrant should get around that problem. And with that, I will wrap it up. It was a big team that it takes to get through this, but especially point out the lead chemist, Kristen and Tyler, and the project lead, Sam Goodchild. And with that, I'll take any questions. Currents goes down. It's even more dramatic actually than the increase in iPSCs that we see. And I don't fully understand that, but that does seem to be the case.
Did you see any signs of inverted U-shaped pharmacology, like you add too much of the compound? What happens?
No, we do not see what I would call U-shaped pharmacology. But eventually, if you push too high, you will get toxicity. So we don't push beyond that at that level.
It's kind of along those lines. I was just curious, in somebody, in a mouse or potentially somebody without Dravet syndrome, do you think there could be therapeutic benefits on inhibitory tone that extend into other indication space?
It's an interesting question, and I wouldn't say I know the answer, but yeah, there have been implications for Nav1.1 and things like Alzheimer's, it would be extremely cool if we could end up benefiting other patient groups. Right now we're pretty laser focused on Dravet, but if we have a clinically enabled compound, then I think our vision might expand.
Thank you for the very interesting talk. And I'm wondering that Dravet syndrome model mice is also difficulty as autistic phenotypes. Have you ever checked the DNA fractions by the
Yeah, that's an interesting question. We have not really dug into the autistic phenotypes in these mice, but with this chow dosing, paradigm, it would be amenable to those kind of experiments, but we haven't gotten to those yet.
And also another question is, you showed the phenotype fractions by administration of the compound in the early stage of the mice. I'm wondering have you ever also tried the late fraction in the adult mice?
Yeah, that's also an interesting question. Once the mice are beyond this kind of critical phase, we have looked in older mice, the mice that survive to about, I think we've looked at about 60 days, and you still see this discrepancy in the LTP between hets and wild types, and the drug does still seem to correct it. But we haven't done a lot of work on those fronts. I think we still have. Yep.
Hey JP, that was a great talk. Really exciting. We've messed around with gating modifiers for 1.2 and 1.6 in the lab as things that are potentiators for those channels. And if you look at those in a pyramidal cell, you actually end up getting more trouble than help. It's almost like the action potential changes from a normal pyramidal cell neuronal action potential into something that looks more like a cardiac action potential, where there's this really long hump. And I think it's because those pyramidal cells don't have enough potassium channels to really control their excitability. Part about BMN cells, by contrast, have a whopping ton of K channels that can control this excitability. And so this might be a case where a potentiator works really, really well. So the question is, what do the spikes look like when you onboard this at a really high resolution?
I don't know the answer to that at a really high resolution. I would say grossly they look pretty normal. However, we haven't really done the kind of experiments that you would do to really characterize the shapes of the action potential. So that's something that might get to the top of our list, but hasn't yet. And then I think Theanne had a question there, unless I answered it. Or
Yeah, JP, I have a question here. JP here. Really, let's talk.
Hey, June.
Can you share how you funded the compounds and where it banned?
We have not disclosed that yet, but I think someone skilled in the art might guess where they bind.
Thanks.
Last question.
I just had a quick question about whether or not, it's kind of two questions, they're pretty short each. One, have you looked at the effect of the drug outside of the outside of the brain? Spinal cord and sensory neurons also express 1.1, and just more so out of curiosity whether or not it's doing anything outside of central circuits. And then the second question might be a little related to this U-shaped question. What happens if you give this drug just to like a normal mouse? If you just potentiate 1.1 basally, especially thinking about like the LTP function, does it have a positive effect, or does it really specifically need to be administered under pathological conditions?
Yeah, it's a good question. Both good questions. And I don't really know the answer to your first question about what it's doing outside of the brain. It is something that I think about, especially when I read your papers. But we don't really know. Now, this motor control issue, we kind of ascribe it to being a brain effect, but it doesn't have to be. And it might be that it actually is partially happening peripherally. I don't know the answer. And as far as giving this to normal mice, if you dose it too high, you will cause problems. We do see that the mutant mice tolerate more drug than the healthy mice, as you would expect. But at the concentrations that I'm showing here, which are beneficial for the dravet mice, it's well tolerated in healthy mice. So we're hoping that we can do studies even in healthy volunteers and get to the levels we want to get to in patients.